Can you hear me?
Yeah, I can hear you.
Hey, sorry for running late. I was coming back from lab. I'm your
That's okay.
co-moderator, and I'll be
We are
adding the
live,
questions to your the
Dan.
Google Drive folder.
Yep.
Yes.
We are live.
Oh, okay.
I'm just going to wait until 6 before we start. So,
Awesome.
perfect. Give everyone a chance to join us. Okay, and just like that, we've hit 6 p.m. my time. So I'd like to thank everyone for joining us here today for the TEDx Black and Immuno Adaptive session. And thank you for joining us in general for Black and Immuno Week. So this week is obviously all about celebrating, amplifying, and supporting the voices of Black people within immunology. And my name is Dr. Faith Iwaji, and I'm going to be your moderator for this evening. Evening for me, I guess, morning, afternoon for everyone, depending on your time zone. So I'm pretty excited to get into the talk today. Today we're all about adaptive immunity and seeing what's going on with the adaptive immune response. But before I start the event, I'd like to take a moment to thank our major sponsors. So we have two major sponsors. We have the Immune Deficiency Foundation and Imperial College London. So like I said before, all the talks today are going to focus on the adaptive immune response, which is basically how our body forms memory against pathogens, and also looking at the implication that this has on diseases as well. So we want to get as many people fully engaged as possible. So please use the hashtag on Twitter. So that is Black in Immuno Adaptive, where all the words are capitalized to try and make it as accessible as possible. So today we're going to have 4 TEDx style talks, and then that will be followed by a Q&A session. And all our presenters will have captions included with their talks to make it easy for everyone to get access to it. And this captioned video will be uploaded on YouTube afterwards, and all transcripts will be made available after 24 hours, and that will be available on the website blackandimmuno.org. So without further ado, I'd like to introduce our speakers. So we have Dr. Annie Ogbe, we have Dr. Azar Gadir, Dr. Michael Okori, and we have Dr. Avi Posey. And our first speaker is going to be Dr. Annie Ogbe. So I'd like to tell her to come to the fore, and I will start an introduction. So Dr. Annie Ogbe is a postdoc researcher at Oxford University. She started her career doing an MSc in molecular medicine and cancer research at the University of Brunel in London, before then completing a PhD also at the University of Brunel, where her research was really focusing at looking at the development of T follicular helper cells and the role that these ERG2/3 genes plays in this role. So to date, her research has really been focused in trying to understand the complex and dynamic roles that these T cells, in particular CD4 positive T cells, make in the development of immunity to infectious disease. So during her postdoc, she started looking at T follicular helper cells and trying to look at the role that they actually play in development of, broadly neutralizing antibodies. And that's during HIV. So she really is a tour de force because the pandemic came along and she took that knowledge and she took that skill set and started looking at the role that lots of cells are making during SARS-CoV-2 infection. So assessing the immune response during that as well. So I would like Dr. Ogbe to take the stage and please present to us your amazing data.
Thanks, Faith, for the kind introduction, and welcome to everyone. So I'm going to be talking about 2 viruses that I've had the opportunity to work with, and that's HIV and SARS-CoV-2. And I'm going to be presenting data from our SARS-CoV-2 work. So I thought, because I had invited some non-specialist people, I thought for their benefit, it would be important to just sort of give them a highlight of what the immune system is, and so that they know what part of the immune system I'm going to be talking about. So basically, when a pathogen first invades the body, the cells of the innate arm would engulf that pathogen and present parts of that pathogen to a CD4 T cell. The CD4 T cell then takes that and then activates a B cell to make them make antibodies. This is what we would call the humoral immune response. But the CD4 T cells can also provide help to CD8 T cells. So these are the cytotoxic T cells, and help them to mediate lysis of infected cells. And all of this would end with pathogen clearance and the development of immunological memory, so that upon reinfection, there's a rapid and robust response. However, sometimes this gets dysregulated and you have a reprogramming of your CD4 and your CD8 cells to this exhausted phenotype where they start to express various exhaustion markers. And I do, my work is mostly centered around the parts squared in red. And I do that in the context of HIV and recently SARS-CoV-2. So why HIV? Well, because HIV still remains a global challenge. As at the end of 2019, there were 38 million people living with the virus worldwide. As at the end of 2019, again, we have had about 33 million deaths due to HIV or AIDS-related causes. The highest disease burden is in sub-Saharan Africa. Thankfully, there is treatment, and we've got about 25 million people currently on ART. However, ART is expensive, adherence is poor, retention in care and drug-related toxicity and resistance still remains a problem. And so this highlights the need for
Dr.
further
Ogbe, just to
work.
let you know, you need to go into presenter mode, and your slides aren't changing either. What can you see? There we go, that's better.
All right, okay, so so like I was saying, ART is expensive, and so there is a need for us to keep on researching on other treatment modalities. One of the treatment modalities we have are the prophylactic HIV vaccine. However, there have been challenges with that, including the rapid mutation of the virus that enables it to escape from detection by autologous neutralizing antibodies. But there's also the diversity in the circulating strain, so that any vaccine that's going to be made would have to be broad enough to target all the subtypes, or would have to make different vaccines for different subtypes. And also, it's likely that whatever vaccine that we're going to make would need to stimulate the various arms of the immune system and induce long-lasting responses, which can be a challenge when you want to engage the B cells, the innate response, and the cytotoxic T cells in one vaccine. What about the cure front? Well, achieving a cure for HIV is proving quite challenging because of the presence of a latent virus reservoir that tends to reactivate upon ART cessation. Can you guys see the next slide now?
Yes.
Perfect, okay. So I started my work in HIV in the BNAP prophylactic vaccine field. And basically there in the lab of Professor Seth Burrow, we're asking a very simple question, which was to investigate the role of the TFH cells in the induction of BNAbs, these broadly neutralizing antibodies, in a subset of chronically infected people that made a spectrum of BNAbs from the low neutralizers to the high neutralizers. And the premise for that work was that if CD4 T cells, well, we know that TFH cells are important in generating humoral immune responses, but were they particularly crucial for the induction of HIV-1 BNAbs that show a high degree of somatic hypermutation, which is another role that we know TFH cells mediate? And if they were, what functions of this or what features of this HIV-specific TFH cells were important? Unfortunately, I'm not going to be showing data from that study today, but that manuscript is in preparation, so look out for it. Something that came out of that work was an optimization of this boosted activation induced marker assay, where basically you have an unactivated T cell that could either be a naive cell or a circulating memory cell that hasn't encountered or re-encountered an antigen. Upon antigenic stimulus from whatever source, natural infection, vaccination, or in the labs, they tend to upregulate these activation markers, and you can use either a single activation marker or a combination of this activation marker to gate on your antigen-specific cells. And that assay was quite nice. We've used it in 2 papers now. One is in preparation, and the other one is currently under review. So I then moved on for the last year. I have been working in BNAbs and HIV cure research, where I'm involved in a clinical trial called the REAL clinical trial, which is basically a randomized trial that investigates the effect of long-acting BNAbs versus placebo on viral control during analytical treatment interruption in HIV-infected individuals that were treated during primary infection. And it's well known in the HIV field, at least in the last couple of years, there have been a lot of studies that have been carried out in macaques and humans that have shown that either a single infusion or combination infusion of bNAbs mediates some kind of viral control. However, what's less known is the immunological mechanism that underlies this, and that's what we're going to be investigating. And that is going to start in 2021, well, recruitment starting in 2021. So now to what I have been doing the last couple of months, really. I have been working on SARS-CoV-2 and during the pandemic. And just some stats. I know it sounds like preaching to the choir, but just some stats. As at the end of last week, we've got 53 million people infected with about, 53 million cases with about 1.3 million deaths. I think there's about 11 million people currently infected. The highest disease burden is in North and South America and Europe. One problem that we have encountered in the field is the cross-reactive immunity to other coronaviruses, which pose a challenge in accurately quantifying and qualifying the SARS-CoV-2 specific immune response. And so it's hampered the identification of immune correlates. So it's quite clear that in order for us to delineate this cross-reactive immunity from SARS-CoV-2 specific immunity, that we needed more sensitive assays. And so with the PIs in the building I work with, they formed the consortium, which we call PEACH, which is Protective Immunity from T cells to COVID-19 in healthcare workers. And together, we all came together to ask the question, can we characterize the SARS-CoV-2 T cell immune response in PCR positive healthcare workers using immunological assays that can differentially capture T cell functions? And there were two main questions that we wanted to address, and that was if the COVID-19 patients and seronegative control showed different levels of responsiveness in different T cell assays. And if they did, can we then use those assays to distinguish people that were previously infected from people that have been infected with seasonal coronaviruses versus people that actually have SARS-CoV-2? So the first piece of data to show you is our ELISA and ELISpot data. On the top panel, on the left-hand side, you have the anti-spike ELISA, where we can show very clearly that you only get an anti-spike response in people that were PCR positive, but not in the seronegative controls or in people in serum that was collected pre-pandemic. Looking at neutralization data, we see exactly the same thing. So only people that were previously PCR positive show this neutralizing antibody levels, and it was significantly higher than the healthy controls. Then we did an interferon gamma ELISpot, which pretty much replicated what we saw with the ELISAs, where you get spike responses to structural and accessory proteins, and to the predicted pool from Alexandra Setter's lab in people that were PCR positive, but not the healthy controls. And when we used our FEC pool just as a control for antigenic T cell response, we found that pretty much everyone responded to it. And what I'm not showing you is ICS data, where we found that this response was mostly polyfunctional. So what we then came up with as well was a way to make our assays even more sensitive. So I have talked to you about the activation-induced marker assay. However, if you culture cells for a longer period of time, you're able to enrich them for antigen-specific cells, and this is called the T cell proliferation assay. So we then use the T cell proliferation assay to address, to then look a bit deeper into this SARS-CoV-2 infected people. And here I'm just showing the gating strategy where we gate on our CD4 and CD8 cells. And you're looking at mitogenic stimulation here, PHA, and you can see that what you're looking for is the downregulation or reduction in MFI of your CTV labeling. And with each division, your CTV stain should split in two. So mitogenic stimulation shows nice CTV downregulation and thus proliferation. With your DMSO control, you don't see anything. This is your media-only control. And then on the bottom panel, I'm showing you representative staining from a SARS-CoV-2 positive person. And you can see that we do get robust responses in both the CD4 and the CD8 compartment. The next thing we then did was to then quantify the SARS-CoV-2 specific immune response in these people using an array of peptide pools from SARS-CoV-2. And what you can see is that in both the CD4 and the CD8 population, when we looked at either the spike protein here split into S1 or S2, or M, NP, ORF3, and ORF8, people that were infected were very much likely to make a response to these proteins or part of the protein as compared to ORF6 and ORF7. So indicating that the T-cell immune response was preferentially targeting this region. On the right-hand side, I've got a heat map to just sort of help you visualize what we see. And you can see that when you look at M and NP, you find this robust response in pretty much everybody. Where it's in gray, it means that we didn't have enough material to test spike, but where we have tested spike always comes up. And then you don't see so much for ORF6 and 7, and it's the same with the CD8 compartment. So what we then did was we had the seronegative control that we had taken earlier in the pandemic, and we tested them with the proliferation assays. And we're quite surprised to see this robust and, huge responses to spike, S1 and S2, but not so much to any of the other SARS-CoV-2 proteins in both the CD4 and the CD8 compartment. But we thought, wait, hang on a minute, maybe these people think they are uninfected, but maybe they had some kind of exposure and they had asymptomatic disease. And because we know that not everybody would make antibodies, we then thought to go back to pre-pandemic samples. So we went back to samples that were frozen down from 2017 up until 2007 to 2017. And we tested those. And again, we found the same thing. So that we had huge responses to S1 and S2 and not so much to the other SARS-CoV-2 proteins. And this was verified again in our lactate assay, which is another sensitive way of looking at T-cell activation. So we then asked, well, can we use the data that we get from the proliferation assay to sort of, really pinpoint what proteins people that are infected would preferentially respond to and what was cross-reactive. And indeed, what we found was that when you look at the S1 and S2 responses, there were no difference between people that were seronegative or people that were PCR positive. However, when you looked at M, N, ORF3, and ORF8 responses, at least you find that there is a significantly higher magnitude of these responses in people that were PCR positive for SARS-CoV-2. Not so much for ORF3 and ORF8 in the CD4 compartment, but certainly in the CD8 compartment, you do see that. So we concluded that responses that targeted M, NP, and ORF3 and 8 were a hallmark of SARS-CoV-2 infection. And so what we then did lastly was to take a cohort of highly exposed seronegative donors that had either been screened for a PCR, some of them had been screened for a PCR and came up negative, or the others didn't get screened. There were 10 of them in the cohort, and we screened them using our assays. So with the early spot, we found 3 of them came up positive. So meaning that they indeed had some kind of exposure to SARS-CoV-2. And with the proliferation assay, in line with the sensitivity of the proliferation assay, what we see is that at least 7 out of 8 of them have got responses that are very much akin to what you will find with people that were PCR positive. And indeed, when we looked at the breadth of the responses in both the CD4 and the CD8, we found it to be significantly broader in the highly exposed healthcare workers compared to the seronegative controls. And looking at the magnitude of the response, certainly in the CD4 compartment, it is significantly higher. So the summary of that study, we found that the detection of cross-reactive T-cell response to SARS-CoV-2 is critically dependent on what assays you've used. Assays with prolonged stimulation time, like the proliferation assay, highlights spike responses in unexposed subjects. High magnitude proliferation and ex vivo early spot responses targeting multiple antigens, including M, NP, and ORF3, are a hallmark of SARS-CoV-2 infection. And memory responses to M, N, and ORF3, we think would provide the way to distinguish recent infection from pre-exposure, or indeed vaccine-boosted immunity. And we can sort of really drill down into the role of this in future protection. So what we're currently doing is we're using these assays to then evaluate the durability of the SARS-CoV-2 induced immune response in natural infection. And then also, it's quite interesting what we find with the cross-reactive immunity. So we want to sort of drill down on that and figure out what the source is, what roles they play, and what are the implications for vaccine design. And just to sort of bring home the two viruses that I have been working with, very recently we got funding for a grant to look at SARS-CoV-2, to look at HIV-infected people that were receiving the SARS-CoV-2 vaccine, the Oxford vaccine, and just sort of characterize their immune response. So this is a single label open arm study with the Oxford candidate vaccine. And the reason for this is because we know that HIV infection provides or increases the risk and the severity of diseases that are very easily prevented by vaccine, and that vaccine-mediated protection is usually impaired in people that are HIV positive. So, for instance, the magnitude and the durability of the flu-specific antibodies is compromised in HIV-infected people. So we've got a cohort of 60 HIV-infected people receiving the vaccine, and we've set a criteria that they had to have an undetectable viral load and also have a CD4 count above 350 cells per mil. And I think it's been determined in previous studies that in order for you to mount a robust response to a vaccine, you need to have a CD4 count that is greater than 200 cells per mil. And we're collecting longitudinal samples from these people. So I have presented this data, but I'm only a spokesperson for a whole number of people. And I would like to highlight the team at the Medawar. We call ourselves Team Science, and there's a picture of everyone here. And of course, all the funding bodies and all my senior colleagues and mentors. So that's it. Thank you.
Thank you, Dr. Ogbe, for that amazing talk. It was really interesting, and I'm sure there'll be lots of questions. So if anyone from the audience has any questions, please put them in the YouTube live feed or include them in Slido or use the hashtag #BlackandImmunoAdaptive. So, and without further ado, I'd like to move on to our next speaker, so Dr. Assaying Gadir. So Dr. Gadir is a director of research and development at Seed Health, which is a microbiome company that is really working to try and use breakthrough science to inform live biotherapeutics or microbial products that can be used to treat unmet medical needs. So Dr. Gadir grew up in London and did her MSc in immunology of infectious diseases at the London School of Hygiene and Tropical Medicine, before then doing a Ph.D. in immunology at UCL, also in London. So she then moved and left the UK and moved to the States and moved to Boston's Children's Hospital as part of Harvard Medical School, which is where she started doing work looking at immunological mechanisms that underlie the role of the gut microbiome. So I'm sure she'll give us an insightful talk, and I'll allow you to take center stage now.
Thank you. So the title of my talk is Microbes and Regulatory T Cells, the Invisible Connection to Reversing Food Allergies. I currently serve as the Director of R&D for a private company, Seed Health, but most of the work that I'm going to present today was actually generated in the lab of my former PI, Talal Shatila at Boston Children's Hospital. So health has traditionally been kind of narrowly defined as the absence of disease. And today I'm going to ask you to shift that perspective a little bit and think of health as an active process. And that's primarily due to a cell subset called regulatory T cells. So regulatory T cells are a potent subtype of the adaptive immune response, and they're capable of suppressing inflammation with extreme precision. So when your body encounters a pathogen, for example, and innate cells rush to the scene, and then they're followed by the more specialized cells, which are the adaptive immune cells. Regulatory T cells are the cells that show up at the scene to break up the fight and tell everyone to go home once the infection has been cleared. Otherwise, the inflammation will persist and start to target self-tissue. And so in conditions marked by chronic inflammation, so for example, autoimmune conditions, so things like lupus or multiple sclerosis, regulatory T cells have been shown not to be able to function adequately. So you get this ongoing systemic inflammation, which results in kind of indiscriminate attacks on self-tissue by other cells. So I just wanted to show here on the right all of the different touch points that regulatory T cells and other cell subsets that we know and we've shown in the literature that they're capable of inhibiting. So actually, before I talk about food allergies, I just want to, in short, kind of summarize that regulatory T cells play a vital role in maintaining immune unresponsiveness or tolerance to self-tissue. And so inhibiting excessive immune responses that can be potentially harmful to the host. So I'm going to tell you about how the gut microbiome is able to activate regulatory T cells, and that has important implications for food allergic responses. And the main take-home message of my talk is that immune exposure to microbes and dietary antigens, so aspects of your diet early in life, is able to activate antigen-specific regulatory T cells and imprint the immune system with long-lived memory tolerance responses, preventing food allergy later in life. So food allergy, it's a major public health concern, and its prevalence has grown dramatically in recent years to affect up to 8% of children and 4% of adults. In food allergy, the immune system perceives food allergens as a threat and will mount persistent allergic responses when encountering it, and failure to induce the right type of regulatory T cells in the gut will result in these inflammatory responses and ultimately anaphylaxis, which is when these infants get constriction in their airway. One key point is that this failure reflects kind of a broader breakdown in immunoregulation in the mucosa, because patients with food allergy are rarely ever allergic to one thing. They often have allergies to more than one type of food. And we know from previous experiments that were done in the Shatila lab that if you transfer allergen-specific regulatory T cells from mice that don't get food allergy, so cells that are induced in response to peanut milk or egg, for example, in mice that can't develop food allergy, you can actually transfer the disease. You can actually protect the mice from developing disease. So studies in both food allergic mice and in humans have shown that regulatory T cells are able to lose their ability to suppress in disease. And so instead of promoting inflammation and instead of breaking up the fight, they actually contribute to the fight and start throwing some punches. So this entire process is driven by this pathogenic reprogramming of regulatory T cells. So what we know is that regulatory T cells in the context of food allergy involve they get the increased expression of transcription factors or switches, such as GATA3, for example, or IRF4. But we also know that T helper, we know that the regulatory T cells themselves develop this Th2 signature, which then continues to funnel and to drive production of IL-4 and IL-13. And so one thing to kind of note here for anyone lay that's listening is that Th2 cells are a type of the CD4 effector T cells that are associated with food allergy. One thing I also want to note here is that previous data from the Shatila lab has shown that if you specifically knock out IL-4 and IL-13 on regulatory T cells, you're actually able to largely reverse food allergy in mice. So we basically have very good evidence that food allergy is mediated by dysfunctional regulatory T cells. So what's driving regulatory T cell failure in the gut? And so at this point, we turn to the gut microbiome and the whole kind of hygiene hypothesis. There's a lot of really interesting suggestive data that supports that both our diet and our environment early in life, around the age of 1 in particular, is able to affect the gut microbiome, which in turn can then affect our food allergic responses. The initial gut microbiota of a newborn is actually seeded from different sources, depending on the mode of delivery. So whether the child is born by vaginal delivery or cesarean, for example. And the gut microbiota from vaginal delivery, the first kind of microbes that will seed in the gut will resemble that of the mother's gut or her vaginal canal, whereas when children are born by C-section, the first kind of microbes that are seeded tend to resemble that of the mother's skin. And so the transition from an exclusive milk-based diet to a solid food one has been shown to be associated with a dramatic increase in microbial diversity, and a lifestyle that is common in developed countries, for example, sterile living and lack of exposure to the outside, seems to be a significant risk factor for then developing food allergy, which again suggests that environmental exposures around that age play a major role in disease development. Living in a farm environment and exposure to a large number of pets in the first year of life has been associated with having more diverse microbiota and is also associated with protection from allergic disease later in life. Other than C-section delivery, antibiotic intake, both prenatally and perinatally, and the intake of medication like gastric acid inhibitors have all been associated with adverse effects to the microbiome, so causing dysbiosis, and then again, also correlating with increased risk of developing food allergies. So experiments on animal models have shown that the gut microbiota is able to modulate the intestine either directly or indirectly by producing metabolites. And we know that the gut microbiota at this point is able to influence the differentiation and the function of regulatory T cells and effector T cells. So in fact, my colleagues at Boston Children's Hospital and myself identified that microbes are actually able to induce a very specific type of regulatory T cell, which can then specifically switch off food allergies. First, we started in the clinic and we analyzed kind of the longitudinal development of the gut microbiota in 154 babies. These babies were enrolled between the ages of 1 to 15 months, and we did 16S sequencing on their fecal matter and followed the clinical progression every 6 months all the way up to 30 months of age. Keeping in mind that kids generally get diagnosed with food allergy around when they start to wean onto solid foods and they start to encounter more diverse dietary habits. So what we found was that food allergic infants actually manifested this kind of early onset dysbiosis. So you see this very specific microbial signature that is associated with food allergic children, but is not associated with children that don't develop food allergies. And you see this around the age of, you see this divergence start to happen around the age of weaning as they're introduced into solids, and this persists into early childhood. So again, this indicates a role for evolving dysbiosis. After we made our human clinical trial observations, we next wanted to see, well, what was the function of this observation? What's the physiological relevance? So we decided to run FMT experiments. So we took fecal microbiota transplantation and we gavaged it into food allergic mice, so mice that are prone to develop food allergies. So what we found was that fecal matter from human subjects with food allergy and fecal matter separately from food allergic mice is unable to protect allergic mice against the development of food allergies. And by contrast, when you transfer fecal matter from healthy infants, and separately when you transfer fecal matter from mice that aren't able to develop food allergies, you can actually protect the mice from the development of food allergies. So we next wondered what in the fecal matter is actually conferring the benefit. So we designed 2 cocktails of bacteria based on the data from the clinical trial. One mix was Bacteroidales, another one was Clostridiales, and we administered it to the food allergic mice. And spoiler alert, we found that it actually, both of these cocktails were able to confer protection. And so we showed that bacteria therapy is able to work as a preventative. So if you give it before inducing food allergy in the mice, but it was also able to reverse disease that's already established. And what was most interesting is that in all of these individual experiments that we ran, we found that as you administered the microbiota, whether that was fecal or bacteria, this was, and you started to see protection from disease, this was correlated with an increase in a subset of regulatory T cells that express this transcription factor, ROR gamma T. So we next wanted we went back into the clinic and we wanted to confirm that ROR gamma positive regulatory T cells in peripheral blood. So just no activation, just taking peripheral blood from food allergic infants and then bleeding the mice. We found that food allergic infants, even compared to atopic infants, so these are kids with eczema and asthma, but no food allergies, have reduced levels of ROR gamma expressing regulatory T cells peripherally. We then went back to the mice and we decided to generate conditional knockout mice. And so we took these food allergic mice, wild type mice, don't develop food allergy. And we specifically knocked out ROR gamma using YFP pre-mice only on the regulatory T cells. And what we found was that these mice were actually able to be sensitized to food and were able to develop food allergy. As a follow-up experiment, we then wanted to see whether monotherapy with bacteria, whether it needed to be a consortia or whether monotherapy could be protective. And we found that monotherapy with a type of Clostridiales, this is a strain, Subdologranulum variabile, that we actually showed was incredibly reduced in children around the age of 1 that went on to develop food allergies. And we showed that actually if you engraft this strain, or actually if you gavage the strain, you can actually both prevent and reverse food allergies in food allergic mice. And again, this was correlated with an increase in ROR gamma regulatory T cells. So in summary, we showed that actually immunoprotective commensal bacteria can act to induce the transcription factor ROR gamma, particularly in regulatory T cells, in order to promote tolerance to dietary antigens. And this mechanism is disrupted by dysbiosis and can be restored by treatment with the right bacteria therapy. So there are multiple ways that we can end up in a situation where a child doesn't have the right regulatory T cells in order to avoid food allergy. And the data indicates 2 key contributing factors. The first one is that the environment matters. So the exposure to different microbes early in life drives regulatory T cell responses, which can inhibit inflammation. So think back to the C-section breast milk data I mentioned earlier. And the other factor actually relates to the antigen-specific nature of food allergic responses. Why are children only allergic to peanut, for example, and not other foods? And so we know that even though these immune responses are systemic, we know that they're food specific. So for example, peanut or egg specific. And a really important unresolved issue here is whether the ROR gamma regulatory T cells that are induced by the microbiota are allergen specific, or whether they're acting in a non-allergen specific manner just to inhibit infectious tolerance. And so previous studies have actually documented that inducing regulatory T cells in the gut, that the regulatory T cells induced in the gut are actually enriched receptors that can be specific to different microbiota. But on the other hand, we are able to induce allergen-specific regulatory T cells in vitro without needing exposure to microbes. So further studies are really needed here to elucidate what's going on with this connection. So what does this mean for food allergies? There seems to be, there seems to be a really important, something special about the weaning window. Something special about the weaning window. It seems to serve as kind of a critically timed window during which the gut microbiota is changing and expanding its composition. So given that a shift in microbial diversity and composition is seen in babies upon the introduction of solid foods or around the time of weaning, we actually propose that immune exposure to microbes and dietary antigens during this weaning window is what's imprinting the immune system with the right regulatory T cell population with long-lived tolerance in order to break up the fights. And concurrent exposure of foods in the gut also induces the formation of these regulatory T cells that are imprinted to both the bacteria and the food. And so the tolerance at this stage may have long-range effects that last well into adulthood, but we do know from these studies that these reactions are reversible. And as we showed in the mouse models, bacterial therapeutic interventions at any age theoretically may be able to reestablish oral tolerance and correct the underlying pathogenic dysbiosis and restore the healthy microbiota. So while future studies are needed to clarify the role of microbes in cellular induction of ROR gamma and the subsequent halt of the allergic cascade, this research could actually shift the way that we treat food allergies moving forward. And feeding children the right microbes and dietary antigens in early windows of development could actually one day prevent the emergence of food allergies altogether. So all of this work was done at Boston Children's Hospital with my PI Talal Shatila. And most of these ideas actually originated with discussions from him. Professor Rima Rashid oversaw all the clinical discussions. Dr. Magali Noval-Rivas was the postdoc before me, and Emmanuel Stephen is continuing the work. Thank you.
Thank you, Dr. Gadir, for that. That was really interesting. I'm sure there'll be a ton of questions about food allergies, and maybe we just need to be giving everyone microbiome and lots of bacteria, but I'm sure there'll be lots of questions about this. So again, like I said, if you have any questions, please add them into the live stream, or please put them on Twitter. So I'd like to take this moment to introduce our next speaker, so Dr. Michael Okori. So Dr. Michael Okori is an MD PhD candidate at the University of Chicago, and he recently completed his PhD in immunology in the lab of Marcus Clark. So he's currently on an MD program, so continuing the hard work, but he is originally from Southern California, and he graduated from the University of California originally with a BSc in biochemistry. He then spent some time on an NIH-sponsored research assistant position, and that's where he started getting interested in the immune system and how it develops. So these experiences shaped his passion for research and scientific discovery, which led him to do this PhD. And his research really focused on molecular mechanisms during B-cell development that promote self-tolerance and antibody diversification. So I'd please like to invite Michael Okori to give us a talk. Thank you.
Thank you for the introduction, Faith, and thank you to the Bio and Immunology organizers for inviting me to speak. So I'm going to be sharing with you all a small portion of the work that I did in graduate school in the lab of Marcus Clark. And so in the Clark lab, we study B-cell development. And as you might know, B-cells are an important and underrated part of the adaptive immune response. And their main goal or main job is to secrete antibodies that can specifically bind to various pathogens that are trying to infect the host. So like viruses, bacteria, and fungi. A crucial aspect of B-cell function is the specificity. These antibodies have to be specific to these different pathogens that they're trying to recognize. And so it's actually quite surprising that the specificity of B cells is generated randomly through a process called VDJ recombination. So when an antibody is constructed, it's made up of two parts, a heavy chain and a light chain. And these are assembled at different stages of development. And within the heavy chain and the light chain, these are composed of even smaller gene segments called VDJ genes. So in the heavy chain, this is made up of a VDJ and a constant portion. And the light chain is just a VJ and then the constant portion. And you can also see there's numerous V, D, and J genes that are present at the various loci. And so in antibody formation, these V, D, and J genes are randomly chosen to be recombined together to form the heavy chain and the light chain. And then these are then paired together, and that then confers the specificity of the antibody. But as you might imagine, this creates a situation where there might be significant risk of producing self-reactive B cells because you can't necessarily control what these antibodies are going to be specific to. And that's exactly what was observed in this wonderful paper by Eric Meffrey and Michelle Newson-Swag in 2003. What they did is they took bone marrow aspirates from healthy individuals, and they were able to get identify B cells that were early in development versus B cells that were mature and late in development. And what they observed is that early and mature B cells here have an antibody specificity that's predominantly autoreactive. And they're showing this here in immunofluorescence staining, showing that these antibodies are specific to nuclear, subnuclear, cytoplasmic, or just cytoplasmic regions in the cell. But another interesting aspect of this paper is they observed that as these B cells are developing as these B cells are going through development, they predominantly lose this ability to bind self-reactive antigens and are predominantly at the end of development non-self-reactive. And so there must be a system that is developed through evolution that would allow for loss of self-reactivity early in development and would cause mature B cells to be non-specific and able to participate in immune reactions. And one of those processes is receptor editing. But before we get into receptor editing, to conclude that they found that 70% of the initial antibody repertoire was actually autoreactive. So this is actually a huge amount of autoreactivity that's early as these B cells are developing. So before I get into receptor editing, I'm just going to start with what normally occurs during B cell development. So we'll start here in the small pre-B cell stage. And this is after the heavy chain has been recombined. And so at the small pre-B cell stage, the light chain recombination is occurring. And the light chain will pair with the heavy chain and be expressed on the cell surface as an immature B cell. This expression of the B cell receptor, the specificity gets tested in a sort of passive manner. And so if this immature B cell binds something in the bone marrow, then, or doesn't bind something in the bone marrow, then it's innocuous and can be positively selected into the spleen. However, in a situation where this newly formed B cell receptor is autoreactive and binds something in the bone marrow, some of these cells will undergo the process of receptor editing. And what that essentially means is this autoreactive B cell receptor will be downregulated from the cell surface, and these immature B cells will return to the smaller B cell pool, where they will undergo new light chain recombinations to try to find a new light chain that can pair with the heavy chain and change the autoreactive specificity. If this is successful, this edited B cell will then progress to the immature B cell stage and then further on into the spleen for further development. However, if this process fails and these B cells remain autoreactive, even though they tried a new light chain, these cells undergo apoptosis. So this is one of the hallmarks that I want to try to get across for receptor editing, is that receptor editing prevents the self-reactive B cells from cell death. As I showed you before in the original paper, 70% of the initial B cell repertoire is autoreactive. So it'd be really terrible if the process of removing autoreactivity was cell death. And so if you're able to keep these cells and allow them to change their specificity, then this process becomes a little bit more efficient. So the second aspect of receptor editing is that it contributes to the diversification of the antibody repertoire. And what I mean by that is that the light chain, both in mice and in humans, there are two types of light chain. There's an Ig kappa light chain and an Ig lambda light chain. Normally in development, especially in the mouse, most of the light chains are going to be recombined here at the kappa light chain. So in the process of receptor editing, when they're trying to find a new light chain to correct for autoreactivity, these mice will try to recombine another allele here at the kappa light chain. And this actually happens quite a bit. As you can see, what I'm showing here is flow cytometry of immature B cells, and 90% of these immature B cells are going to be Ig kappa positive. But every so often, recombination at Ig kappa fails to correct autoreactivity, and these mice will or can progress to the Ig lambda locus. And here in the mice, the Ig lambda locus is quite restricted with only 4 products that are known. And these can be used to then help tolerize autoreactivity. But what's interesting, and so yeah, you can see that there's only around 6%. But what's actually interesting is that in mouse models, in which these mice have a fixed specificity for an autoreactive antigen, they end up showing that a lot of these mice that make it out of the bone marrow end up using the lambda locus. And specifically, lambda X here has been shown to be quite good at correcting original specificity to double-stranded DNA. And then we also know that in humans, the representation of Ig lambda is somewhere between 30 to 40% of the B cell repertoire. But because of the low representation of Ig lambda in mice, we don't actually know what is involved in the regulation of Ig lambda recombination and the regulatory mechanisms that are involved in failure at Ig kappa during receptor editing and then progression to Ig lambda. So that was my question during my PhD, is what regulatory mechanisms are necessary for receptor editing and Ig lambda recombination. So to start, we had to take a step back and figure out, well, what are the ways that you can get an Ig lambda positive B cell? And one of the ways which I described here is receptor editing. So you can start with an Ig kappa positive B cell, and if this cell is autoreactive and remains autoreactive for the subsequent Ig kappa light chains, would have to undergo receptor editing to use Ig lambda locus, which would hopefully not be autoreactive, so these cells can go to the spleen for further maturation. But the other process is you could fail kappa recombination. As I mentioned earlier, you do have this random joining of a V and a J gene, and sometimes this process can be error prone. So you might end up getting failure at the kappa locus. And so you might only be able to recombine successfully at Ig lambda. And so we thought that if we could figure out what the molecular mechanisms that are involved in these two different trajectories of Ig lambda B cell formation, we might be then able to identify, well, what are the unique processes that are occurring here in receptor editing that make this possible for these cells to do this. And so to understand these different processes, we went ahead and obtained 2 mice that we think recapitulate these different trajectories of Ig lambda recombination. So the first is the kappa macroceph mice. And these mice possess a transgene that expresses a single variable chimeric membrane-bound antibody against all Ig kappa, against the light chain Ig kappa. So essentially in these mice, as these B cells are developing, any B cell that expresses Ig kappa initially will be rendered autoreactive. And so this receptor will have to be downregulated and then undergo receptor editing to use Ig lambda locus in order to progress out of the bone marrow and into the spleen. And then in the second, model, this is a KDOL model. These mice have a genomic deletion of the J gene segments at Ig kappa. So the only functional light chain in these mice is Ig lambda. And so this would be a model of undergoing recombination to Ig lambda without this autoreactive intermediate. And so here I'm showing you some flow cytometry where I'm staining for the heavy chain here on the y-axis with IgM, and then the light chain here on the x-axis. And it should just show you that this is a fairly clean model. So again, in wild-type mice, you're gonna have a huge proportion of them being Ig kappa positive. Both the KDOL and the KMAC, they don't have any Ig kappa positive B cells. And we've shown that, again, all these light chains are gonna be Ig lambda. And so for the experimental design, what we did is we took bone marrow from wild-type, macrocells, and KDOL mice, and then we sorted the small pre B cells and immature B cells. So again, the cells that undergo light chain recombination, and then the cells that are essentially testing the specificity that's resulting from the pairing of the heavy chain and light chain. And then we submitted them for a number of sequencing technologies, but I'll be going over a little bit of the RNA-seq data that we saw to get an idea of what are the genes that are differentially expressed, and then also what molecular pathways might be enriched in one process versus the other. I'm going to summarizing some of that data here using the principal component analysis. And so this is principal component analysis of the RNA-seq data. And in principal component analysis, it's a way for you to understand the variation in gene expression in 2 dimensions. So that's to say that samples that are closer together are going to have less variation, or going to be more similar to each other than samples that are further apart. So a couple of things that I want to point out here is that the small pre B cells, regardless of genotype, are fairly apart from the immature B cells here. And this is something that we would expect because these are 2 different stages of development. What was particularly interesting was that the small pre B cell stage, transcriptionally, there wasn't very many differences between the different genotypes. However, at the immature B cell stage, there's quite a bit of difference. And so to focus us here, I'm going to show in the next slide the results from the differentially expressed genes between the KDOL immature B cells and the macrocells immature B cells. So here's a volcano plot summarizing that data, where on the y-axis, we're looking at statistical significance, and on the x-axis, this is fold change. So in total, we saw 2,000 differentially expressed genes were upregulated in the macrocell immature B cells, and then 2,200 so genes were upregulated in the KDOL immature B cells. But one particular gene, which I'll highlight here, and we'll focus on this for the rest of the talk, is CXCR4. And this was upregulated specifically on the macrocyte and mature B cells. So CXCR4 is a chemokine receptor, and its signaling pathway has been implicated in many aspects of biology, including gene transcription, survival, and chemotaxis. However, in B cells, traditionally, the role of CXCR4 has mainly been thought to be chemotaxis, as in expression of CXCR4 is what allows cells to home to the bone marrow and to stay in the bone marrow, and to be trapped in the bone marrow. But we recently published, I think this came out last year, that CXCR4 signaling actually directs Ig kappa light chain recombination and some other aspects of B cell lymphopoiesis. And so this is a heat map from this figure where we're showing here small pre B cells. Again, these are the cells that are undergoing light chain recombination. And we're showing gene expression from wild type and from B cells that have specifically knocked out CXCR4. And you can see there's quite a few genes that are dependent on CXCR4 expression in order to be either downregulated or in order to be upregulated, really enforcing the idea that CXCR4 is signaling is important for B cell function and maturation. And then also in this paper, we showed that CXCR4 is important for Ig kappa light chain recombination. Another paper that had implicated a role for CXCR4 biology is in B cells is this paper here, which showed that in order for B cells to continue in development, I mentioned that they have to leave the bone marrow. And that process involves downregulation of CXCR4. But in this paper, they also had a really interesting experiment where they took B cells from either wild type mice or from mice that had a fixed B cell receptor, so they're only specific to one antigen. So there's B1A and there's MD4 mice. And what they did is they took these immature B cells and they stimulated the B cell receptor with either anti-IgM or with the cognate antigen. And what they saw is that after 6 hours, CXCR4 was upregulated on these cells in the various mice. And what they concluded from this is that in this setting where immature B cells are essentially, again, testing their specificity, if they end up binding something, if this receptor signals, they upregulate CXCR4, presumably as a mechanism to trap them in the bone marrow and prevent them from going out into the spleen. And we thought that this could also mean that this trapping of them in the bone marrow would also facilitate the ability for these cells to undergo receptor editing and then change their specificity, so they might eventually be able to leave the bone marrow and continue maturation. And so us observing CXCR4 in our model of receptor editing really had us asking whether or not CXCR4 had a specific role in receptor editing and maybe might be controlling some of the recombination events that are happening there. So to answer that, we use this CXCR4 MB1 Cree mouse, which is a CXCR4 deletion specifically in B cells that we used for our previous paper, and we crossed it to our macrosoft mice. And we generated macrosoft mice that has CXCR4 deleted or was a flox control. And here I'm showing flow cytometry for the different stages of B cell development. Our focus is down here on these graphs. And we show that in early development, so the pro B cells and large pre B cells, in these stages, heavy chain recombination is occurring. The loss of CXCR4 did not seem to impair the formation of these subpopulations. However, starting with the light chain recombination, progressing further, you saw loss of these other stages of development, suggesting that CXCR4, the loss of CXCR4 was impairing the ability for these later stages of development to occur. But again, a crucial question is, does CXCR4 regulate Ig lambda B cell formation? And so to answer this, I'm showing here flow cytometry of the kappa light chain and then the Ig lambda light chain. And in black here is the flox control, and then in blue is the CXCR4 knockout. And what you can see here in this orange quadrant is that cells that have lost CXCR4 have lost the ability to express Ig lambda on the cell surface, suggesting that Ig lambda is important, or excuse me, that CXCR4 is important for the formation of Ig lambda positive B cells. However, I told you earlier that in most of these mice, they need to recombine Ig kappa first before they progress to Ig lambda. So this result that we're observing could be an indirect result of a defect first at Ig kappa before progressing to Ig lambda. So in order to rule this out, we decided to do intracellular staining for the presence of an Ig lambda positive B cell, with the idea that even in the context of CXCR4 deficiency, Ig kappa recombination could be fine, and this receptor could be expressed on the cell surface. But because it's on the macrocell background, Ig kappa would be autoreactive, and so this would be downregulated. However, when you lose CXCR4, you have the inability to then transition from recombining Ig kappa to Ig lambda. So that would then explain the reason why we don't see any Ig lambda positive B cells when we delete CXCR4 in the context of a macrocell. And that's exactly what we saw. So again, here in black is going to be the flox control, and in blue is going to be the CXCR4 knockout. And so I'm showing you intracellular staining for the heavy chain, IgM, and then for the light chain, Ig kappa, Ig lambda. And as you can see, loss of CXCR4 does not impair the ability of Ig kappa to pair with the heavy chain. So these are expressed at equal proportions. However, when you lose CXCR4, you have an inability to form Ig lambda recombination. And so that means that in the setting of CXCR4 deficiency, there's a selective loss of Ig lambda recombination. And so again, this was a small portion of the work that I did in sort of understanding receptor editing and Ig lambda recombination. We were able to show that there's profound differences in chromatin accessibility and also transcriptional state when these cells are undergoing receptor editing. And I showed you a little bit here about the role of CXCR4 in controlling DNA recombination that occurs during this state. So with that, I'll go ahead and conclude and thank, again, with everyone in the Clark Lab. Was a really great experience. And also a lot of other people who contributed to this work that I wasn't able to show. And with that, I look forward to your questions in the question and answer section. Thank you.
Thank you for your talk. That was really interesting. I guess I've always thought of CXCR4 just for localization. So yeah, quite insightful. Okay, and last but not least, I'd like to introduce our last speaker. So that's Dr. Avery Posey, who is an assistant professor in the Department of Systems and Pharmacology and Translational Therapeutics. He's also a member of the Parker Institute of Cancer Immunotherapy and Research Institute at the Philadelphia Veterans Administration Medical Center. He has 2 bachelor's degrees, one in biochemistry and one in bioinformatics, both from the University of Maryland. And he did his PhD in genetics at the University of Chicago. So the Posey Lab's work really focuses on understanding novel cancer therapies that can genetically alter a patient's own T cells to improve the ability of their immune system to fight cancer. So I'd like to take this moment to give you the stage. Thank you.
Thank you. So I like to start my presentation with this fact, that cancer is the leading cause of death in the United States and the world. And there are reasons why our immune system fails to recognize and eliminate cancers. Those in particular are because there may be a low abundance of targetable mutations in the tumors. And essentially, the tumor is invisible to the immune system, recognized as normal tissue. There may be a low abundance of tumor-specific immune cells able to do that work of eliminating the tumor. There are also mechanisms that the tumor uses to suppress our immune system. And so because of those factors, decades worth of research has developed new technologies in order to modify our immune cells. And one of those technologies is called a chimeric antigen receptor. This uses a method of genetic engineering. In this case, we use a lentiviral vector to express into a T cell a new receptor. And this receptor is special because it allows that T cell to then recognize a tumor cell based on the expression of some molecule on the cell surface of that tumor. And when recognizing that tumor cell surface marker, T cells activate, they kill, and they proliferate. And this is now a drug that is FDA approved for multiple B-cell malignancies, and we hope in the future for multiple other cancers as well. And the way in which this occurs is the patient comes into the clinic and T cells are removed through blood donation. They are then genetically modified in the laboratory, expanded into large numbers. So to get over that hurdle of a low number of tumor-specific T cells, we can generate billions in a lab, and then reinfuse that into a patient, then to target that patient's cancer. And I told you before that that molecule is really special. It's called a chimeric antigen receptor, and we use the acronym CAR for short. And it includes factors that come from multiple adaptive immune cells, including B cells. The extracellular portion is generally derived from an antibody, which B cells produce, and the intracellular portions of that receptor that is expressed on T cells combines factors that are important for T cell activation and effector functions. And when we look at the utility of those CAR T cells in a patient clinically, we see that in a patient with B-cell leukemia, one day before infusion of these genetically modified T cells, notably the patient's own T cells, there's a large number of CD19 positive B cells, leukemic B cells in that patient. And 1 month after infusion, we see the complete elimination of those cells. These genetically modified T cells have the ability to completely eradicate a tumor. But because of a lack of tumor-specific cell surface markers on multiple cancers, we need a nuanced approach to drive this forward to safely target solid tumors. And so to do that, my story starts back in the 80s with a scientific discovery that tumors express truncated O-glycans. And so what are glycans? These are sugars that are usually incorporated into our proteins as well as our lipids. And how does that occur? Well, for O-glycosylation, on a protein's serine and threonine residues, one sugar called GalNAc, or N-acetylgalactose, is added to a serine or threonine by 20 different enzymes. And they recognize these proteins to add this sugar based on some sequence specificity. And then there's one enzyme that takes the next step called C1-GAL-T1, or T synthase, and it adds galactose onto that GalNAc to form what's called the core-1 glycan, or T antigen. This is further elongated normally. That enzyme has a chaperone protein that's required for proper folding in the ER. And if you lose either of those antigens, you end up not elongating your O glycans past GalNAc, and that residual GalNAc is then trafficked to the surface on your protein of interest, and that's called the TN antigen. This can also be silated with a sialtransferase to form another abnormal sugar called sil-TN. And that means that there's an abundant amount of proteins present on the surface of a tumor cell that would normally, in normal tissue, in a normal cell, be highly glycosylated. But in cancer, it's not. And it has these truncated O glycans. That means that we have the ability now to recognize those truncated O glycans with special reagents. And reagents, by reagents, I mean mostly antibodies. And so these are antibodies that bind to glycans, that bind to demasked peptides, proteins that have lost their glycosylation, or a special antibody that we're really interested in is one that binds to the combination of both a protein sequence as well as that truncated sugar. So it has a requirement for both. And this antibody that I'm mentioning is called 5E5. This binds to a TN antigen found on the protein MUC1, and we call this antigen TN MUC1. TN MUC1, as identified by the 5E5 antibody, is present in multiple malignancies. And I'm showing you here at one section from multiple tumor microarrays, bright expression of TMF1, and that bright expression is characterized by that brown stain in multiple solid tumors, but also present in some hematopoietic malignancies like multiple myeloma. And so we think that by targeting TNMUC1, by targeting these alternative glycoforms, we may be able to target tumors that have low mutational burden, where our T cells are not naturally equipped to target because they don't recognize these sugars. They don't recognize these abnormal glycans. And then as a aside, you may be wondering, well, what does TMUC1 look like in normal tissue? Well, it's almost not present at all. When it is present, it's present in the Golgi. That's because this is a initial sugar that makes it to an elongated sugar. That all happens in the Golgi. And so you will find that in normal tissue in the Golgi, but never at the cell surface. And we've looked through multiple tissue sections from multiple individuals and have not identified cell surface TNMUC1 on normal tissue, but you will find that in abundance on the surface of a tumor. And so we utilized that antibody called 5E5, and we generated one of these chimeric receptors that targets TNMUC1 using the 5E5 antibody, and we expressed that in T cells. And to test that, whether or not that those T cells work, we use an animal model in which we give mice, 40 mice in this case, a tumor. This is a Jurkat T cell leukemia. It's a cell line that's, in this case, been made bioluminescent, so we can visualize the tumor in the mouse by looking at this firefly luciferase. And one day after this image was taken, we treated these mice with either PBS in group 1, with T cells that have not been genetically modified, they don't recognize a tumor in group 2, with T cells that were genetically modified with a chimeric antigen receptor that does not target this particular tumor, so this one case targeting CD19, or with T cells targeting TNMUC1. In this case, we gave 2 separate doses. And what I think that you'll be able to appreciate is that in our control groups, group 1, 2, and 3, there isn't a reduction in tumor burden that's sustained in these mice. However, in groups 4 and 5, you do see a loss of tumor burden, and we have an enhancement in the survival of the mice as well, suggesting that these TNMUC1 CAR T cells can eliminate T cell leukemia in vivo. We also assess these mice because the process of elongating O glycans is conserved between mice and humans. We assess these mice for localization of T cells and normal organs. We don't find any. We assess them for toxicity to normal organs. We did not find any. That suggests to us that this is an approach that may be safe in humans. But we also wanted to know if we were to target this abnormal glycan in a normal tissue, to target a tumor surrounded by normal tissue, would the T cells be able to recognize the abnormal glycan, but not the normal tissue? So to do that, we generated a tool, JERCAT cells genetically modified now to re-express the chaperone protein that it lacks, called cosmic, which causes it to lose expression of TMUC1. You can see the difference from here to here. It loses expression of TMUC1, but they gain expression of CD19 because now we've transduced these cells with the CD19 marker. So those JERCAT cells become CD19 positives. And so we label these cells TN positive and TN negative. If we culture them alone in the well, we can identify that TN positive and TN negative cell based on some cell staining. If we incubate those T cells, those T cell leukemia cells with non-transduced T cells, so T cells that are not targeting the tumor, there is no decline in the tumor that we can observe. If we utilize either of the CAR T cells, the ones targeting CD19 or those targeting TNMUC1, we can specifically eliminate either the TN negative cells, which are CD19 positive, or the TN positive cells, which are CD19 negative. So that suggests to us that our T cells can differentiate a hypoglycosylated tumor cell from a normally glycosylated cell, even within the same microenvironment or the same tissue. And that's encouraging. We've then tested these T cells against a panel of tumor cell lines, some leukemias, some pancreatic cancers, some breast cancers. We've also tested them against some ovarian cancers, some lung cancers, prostate cancers, and we see that there is reactivity to many of these cancers. And in the model that I'm showing you to the bottom is a representative mouse from one group treated. Both mice in this case are carrying a metastatic pancreatic cancer. And one's treated with the control CAR T cell that does not target the cancer, the CD19 BP zeta. And then another mouse in this group is targeted with TMUC1 specific CAR T cells. And you can see after 3 weeks of just 3 weeks of the CAR T cells in these mice, the tumor is eliminated. And these mice that are treated with TMUC1 CAR T cells survive 100% out through the range of the experiment, which suggests that these TMUC1 CAR T cells are effective not only against this T cell leukemia line that I showed you previously, but against multiple cancer histotypes. And so that's really encouraging as well. And so we're really interested to see whether targeting this specific glycoform that's present on the surface of a tumor cell will lead to loss of tumor burden and clinically. And so we have a clinical trial that's open now, targeting 6 different indications: non-small cell lung cancer, ovarian cancer, fallopian tube cancer, triple negative breast cancer, multiple myeloma, pancreatic adenocarcinoma. And this is sponsored by a Philadelphia-based biotherapeutics company called Immuno. Additional engineering that my lab does is also focused on glycans. One project in the lab that postdoc Tiffany King has worked on is to engineer T cells with an enzymatic activity that cleaves off the surface of the tumor cell a sugar called sialic acid. Sialic acid, importantly, is a ligand for an inhibitory immune receptor, a family of which are called Siglecs. These are expressed on some are expressed on T cells, some are expressed on B cells. They're also expressed on innate immune cells, NK cells and macrophages and dendritic cells. And the purpose is to shut down an immune response, to inhibit that response when contacting sialic acid. And so our approach is to express this enzyme that's naturally found intracellular in a cell. It leads to breaking down of proteins and glycans. When we express this enzyme on the surface, we can see that these T cells now can cleave off sialic acid on the tumor cell, as shown to the right. We can cleave off sialic acid on the tumor cell, but does that enhance the efficacy of those T cells? And when we combine them in a in vitro assay, an in vitro killing assay with NK cells at a low effector target ratio, we can see that indeed it does, that our TNMUC1 CAR T cells, when added with NK cells and the combination of the sialidase enzyme, either added exogenously or expressed on the surface of the T cell, all leads to synergy cooperative killing, and elimination of the tumor cell. Now, what's important is that when we don't express a chimeric antigen receptor on the T cells, when T cells don't target the tumor, that cooperative killing is not observed. And so that tells us that two things. The CAR is very important. Activated T cells are very important for generating synergy from NK cells, but also that CAR T cells can be utilized to reactivate or enhance the immune response from innate immune cells against a tumor. And so with that, I'll leave us with one statement that our immune system can be trained through ex vivo genetic engineering and expansion into living drugs that are capable of distinguishing cancer cells from normal tissue, that can eliminate cancers based upon differential glycosylation, which is a novel concept that I think that we are working through a proof of concept that this approach, while observed as a biomarker in the 80s, can be utilized, can lead to drugs that may eliminate cancers, at least in mice and hopefully in humans, and that we can alter the tumor surface to enhance immune responses to cancer. So with that, I thank you for your attention, and I also will await any questions. Thank you.
Thank you, Dr. Posey. That was a really interesting talk. And with that, I'd like to open up the floor and invite all our speakers to share their screens again and come on board for the Q&A part of the session. So I'd like to invite the audience to keep posting any questions if they're interested. And remember that you can ask questions about the science, but also about people's careers and how they've got to where they are today. So we've got actually quite a lot of questions that have come through. So I thought I'd just start by asking a few questions to Dr. Ogbe. So there was quite a lot of interest in the COVID-19 element of your work. So one of the questions that was quite highlighted was, can you please speculate on the sources of the spike protein reactivity in uninfected controls?
That's a very good question, actually. So we think that it is due to responses that are targeted towards the seasonal coronaviruses. And I think there have been some papers that have been published regarding that, but I wouldn't say that that is exhaustive. It could be from, other antigenic sources. It could be from other pathogens. It could be from the microbiome. I don't know. We're still trying to drill down on that. But certainly, I think there was a paper from, I want to say Shinkrati's lab and Alexandra Setti, but I'm not completely certain now. But yeah, that's what we think. We think it might be from the seasonal coronaviruses. And yeah. So
more of like a cross-reactivity.
Yeah, exactly. So yeah, and we all know that the exposure to the seasonal coronavirus is pretty much universal. So it's not surprising that a lot of people make that response.
Okay, yeah, that's really cool. I'll just throw you a second question while we're with you. So do you have any opinions on the similarities between S1 and S2 T cell responses in the context of the coming spike-based protein vaccines?
What I can say with regards to that is, I think the Oxford vaccine, the paper in the Lancet did show that when they tested for antibody responses in the people that were vaccinated versus people that had natural infection, they made antibodies to the same level. And when they also tested for T cell responses, they made T cell responses. And what was quite nice was that the response at baseline, and this is measuring, I'm looking at it via interferon gamma ELISpot for the T cell responses. At baseline, they could not see a lot. There was barely any response at baseline, which was what we saw as well. Using the ELISpots, we couldn't really see any responses in the seronegatives. However, with the prime and the boost, longitudinal samples from, across time anyway, they were able to show a nice, increased kinetics in the T cell response. So it will indicate that the vaccines with the spike is in a way boosting your spike response. How this would play out, I think it's something that we still need to watch.
Okay.
This.
Yeah, so we don't know yet specifically between S1 and S2 targeted specific T cells.
No, I think the vaccine is designed in such a way to cover the entire S protein. Was that the question? Did I answer your question?
Yeah, no, you did. I was just probing. The person asked about similarities between the S1 and S2 T cell responses.
Yeah, so the vaccines are designed to cover the entire S protein. However, we know that we think that the cross reactivity would be more focused on the S2 part, because that part is where you get a lot of conserved amino acids between the coronaviruses.
Okay, cool. Thank you. All right. With that, I'll move on to some questions to Dr. Gadir. So there were quite a lot of questions that came in about the food allergy. But one of the questions that came up was, do food antigen specific B cells persist after tolerance is established by providing a tolerogenic microbiota?
Yeah, that's a really, really good question. So what we found in our study was that we stained fecal matter for IgA and for IgE to look at what's going, just as a proxy measure of what's going on with the plasma B cells in the Pius patches. What we found was IgA producing plasma B cells are associated with tolerance, and IgE is associated with food allergy, which I guess makes sense. And so our answer to this is yes, but it's as part of our follow-up study, we're looking into exactly what's going on with the tolerogenic B cells.
Okay, cool. And another question was, do you think there's any exposure to the ideal condition of microbiome, so microbial and antigen, that could induce tolerance later in life, or is it likely to only be used as a preventive measure? Sorry.
So in our mouse work, we actually showed that if you induced, in our 8-week food allergy model in the F709 mice, if you induce food allergy and then you treat with the microbes, you can actually reverse the disease as well. I think that there is some preliminary kind of clinical data which shows that if you are going to correct for a dysbiosis later in life, then you're probably going to need to use antibiotics as well to create a niche for those microbes to take hold. But in answer to that, our mouse studies suggest that actually you can reverse food allergies and induce tolerance later in life by introducing the right microbes or intervening later.
Okay, cool. Perfect. And Dr. Okori, someone asked, is CXCR4 expressed at similar levels in B1 and B2 cell subsets?
Okay, that's a good question. So I haven't looked at B1 subsets to see if there's a difference in CXCR4 expression. But if I had to wager a guess, I'd probably say no, based on where the B1 and B2 cells are. B1 cells are usually in the peritoneal cavity, so they're not in the bone marrow or in the blood. And so I'd imagine that CXCR4 expression would be lower on those cells than in the B2 cells.
Okay, cool, perfect. And is there a defect in Ig lambda recombination after deletion of CXCR4 due in part to premature exit from the bone marrow?
Okay, that's another really good question. So I don't think so. The cells that we analyzed were cells that were found in the bone marrow, and so you can also like differentiate between the cells that recirculate and mature cells that can come back to the bone marrow based on B220 surface expression. And so we don't think that this is an effect of premature egress from the bone marrow when you lose CXCR4.
Okay, cool. Yeah, okay, perfect. Thank you. And Dr. Posey, we've got a few questions for you. Were there any adverse effects seen in the mice with the 5E5 CAR T cells, namely anything like a cytokine storm, giving the robust anti-tumor response?
Sure. So our mouse models, which in this case is an NSG mouse, are not really set up to evaluate cytokine storm and cytokine release syndrome, which has been associated with CD19 CAR T cell therapy and BCMA CAR T cell therapy, mainly because it requires an interaction between T cells, the cytokines produced by T cells in myeloid cells, macrophages, and we don't think that crosstalk is happening between the human T cells and the murine myeloid cells in this case. And so so no, we didn't observe that, but our model isn't really set up for that.
Okay, cool. And does the tumor burden affect the efficacy of the TMUC1? And have you tried a solid tumor like B16?
So the pancreatic cancer model that we use is a solid tumor.
Solid tumor.
And we don't think that tumor burden affects the efficacy in this case, although we haven't really explored that.
Okay. Is there maybe a difference in expression in the different types of glycosylation amongst different cancer types?
Sure. Yeah, we don't think that the TMF1 antigen is ubiquitous throughout the tumor, and that's interesting because even in our pancreatic model, this is a cell line, but even in that cell line, the TMF1 is not ubiquitous. The tumor, I actually took this part out of my slide, but the tumor is heterogeneous in its expression, but we eliminate the entire tumor. And so we have a paper that came out earlier this year in collaboration with Hans Schreiber from UChicago that suggests that T cells targeting TN glycopeptides may be more promiscuous than those targeting proteins alone, that they may target other TN peptides. But if we assume that TN antigen is an abnormal antigen to be expressed on the surface of a cell, this still gives us a cancer-specific tool or therapeutic to target the entire tumor.
Yeah, no, very interesting. Okay, we have quite a lot more questions. Cool. Another question for Dr. Gadir. What causes the onset of food allergy later in life?
I'll give you my answer is events that can cause dysbiosis. So typically you'll find that people who get take a lot of antibiotics for an infection will find that they develop random food allergies after. Food allergies are also associated with the development of autoimmune conditions. So in answer to that, I would say any event that disrupts your microbiome is associated, and that also includes viral infections. Like we're seeing some reports of food allergy emergence with COVID now. So any kind of major event that would disrupt your microbiome.
Wow, so quite varied, the causes.
That's what I thought. I was like, I could.
Yeah. I feel like that's often the story with allergy. It's like, there's many causes.
Yeah.
So I know that you mentioned the induction of your raw gamma T regulatory cells, but is there any efforts to, competitive advantage? So there's any competitive advantage to commensal bacteria implicated in reducing food allergy?
Yeah, this is a really excellent question. So this kind of touches on what's next, right? How does the switch happen to does it happen within the Tregs to ROR gamma? The answer to that's no. We know that it's a distinct lineage of peripheral Tregs that are being induced that express the ROR gamma. These aren't thymus derived, but this is one of the hypotheses that the follow-up postdoc is testing is it that the ROR gamma regulatory T cells are then affording a competitive advantage for certain bacteria to come in, and then you get this feedback loop. So I don't know the answer to that, and that's something that we are testing.
Okay, cool. Very interesting. Okay, lots more questions. Dr. Ogbe, are you seeing an increase in the frequency of viruses with pandemic potential? Are we seeing an increase? Oh, you're on mute. It had to happen at least once in this thing.
Okay, I'm sorry about that, but I was saying that I would say that that's very closely related to elements of, virus factors as well as elements of the environment. So with things like deforestation, we're seeing a loss of certain species due to their habitats being destroyed, and the viruses that would have existed in this species are now making zoonotic jumps to humans. So I would say possibly we may see a bit more in the future, and that would be related to sort of environmental changes and zoonotic jumps from this viruses that shouldn't be, that wouldn't normally exist in human populations. So yeah.
Yeah, and I think if you take that in combination with our lifestyles of moving all around the world, it makes it much easier to maybe jump as well.
Yeah.
I was just wondering, in general, in the light of COVID-19 and the pandemic, is that something that everyone's work has obviously been impacted in some way or some form, but has it changed your outlook to research or immunology in general? And maybe a question to all the panel.
I think if anything, it's made me realize how important what we do is, and how much, you can't really ignore certain diseases because, we tend to focus on, the big players, and certain areas are less funded, but we've had our entire lives pretty much put on hold, the economy crippled in the last year by, COVID-19. And thankfully, there were people who have been doing research against all odds, and that's why we're able to get to the point where we got to with the vaccines. But it's really made me realize that what I do is important. And if I don't do what I do, I can see the impact of what would happen, So yeah, I think it's made me appreciate immunology a lot more.
Yeah, definitely, yeah. Maybe Michael next.
Sure. So I'll definitely say that with the COVID-19 and the way that it's affected science, I think that the perception of scientists and for like the layperson, someone who's not in science, I think it's made me really be cognizant about how you communicate science and make sure that it's something that's clear and something that people who don't understand can trust in it. I think, especially in the States, a lot of the issues where people just didn't know where to believe and didn't know where to go for the truth, essentially, for what the science actually meant. So really just focusing on being clear and being honest about the limitations, not overpromising, things like that. That's kind of what's come to the forefront of my mind with everything that's been going on.
Agreed. Maybe Dr. Posey?
I was actually just going to say exactly what Michael said, that I think that the importance of factual information and scientific communication has become really clear.
Mm-hmm.
Yeah, I would agree as well, is realizing how important the communication piece of it is. Appreciating that after this year, most people know what immunology is, and I think they know what we all do. I think for the first time in my life, some of my friends were like, oh, okay, like, I get it now. But the other thing about COVID that I found quite interesting was, as the immunology of the disease trickled out, being able to kind of map my own thinking of it, if that makes sense, like having bits and pieces of information come in and realizing like how much, like as immunologists, you have like independent thinking and you're passing it all together in real time. I've enjoyed that process. I thought that was as awful as it's all been.
Yeah, no, I entirely agree. There's something about, honestly, I've been telling people I know about T follicular helper cells for about 5 years. And suddenly everyone knows what a T cell is and a B cell and an antibody. It's incredible. And I think like on that subject, it's the idea of like what actually got you guys all started on your immunology journeys, because obviously we're all here. This is a celebration Black in Immuno. Like, what made you think, you know what, immunology is for me, or did you just stumble across it, or yeah, what's your story? Anyone can take the floor, whoever feels the urge.
I'll start because I'm not an immunologist. I'm a
Oh,
geneticist,
very good.
and my interest has always been in gene therapy. And so I love the idea that we could treat disease, improve the human condition by manipulating our genes. And that at the time that I started my postdoc, that was happening rapidly in clinical translation and T cells. And so that's what drove me to this field.
I can go next. So for me, I think it was the opportunity to discover. So when I started my PhD wasn't really focused on TFH cells at the very beginning, but then as we kept on working on this mouse model to sort of elucidate what was happening in the mouse, we found that they had this atypical germinal centers, and that's what led me down the path of TFH cells. And then I think the field was like maybe 5-6 years old, so the literature wasn't huge, and it was just very, very exciting. And there was so much to discover. There is still so much to discover. So I was just really, really interested in those cells. And then I think I've always been interested in HIV because I grew up back in Nigeria, and it was a big deal when I was growing up. It is still a big deal, but no one really understood the science. And so I had this opportunity to, do something that is exciting, something that, gives me this daily challenge, something that keeps me motivated, and work on these exciting things as well. So I could merge TFH cells in the context of BNAbs and then, study viral infections in the context of HIV. And I thought, you couldn't ask for anything more, really.
Yeah, 100%.
I can go next. Oh, go ahead, Michael.
Okay. Yeah, for me, I think I landed in immunology pretty late in college. So I had a personal friend, first family member, get diagnosed with an autoimmune disease. And so at that point, I didn't know anything about it. So that was really kind of the push to start learning about the immune system and how everything works. And then I got really fascinated by the science. So I kind of just followed my path and just sort of trying to understand the immune system, and then also learn a little bit more about health disparities within autoimmune diseases within African Americans, and the fact that a lot of African Americans don't do well when they're diagnosed with autoimmune diseases, and especially the treatments that aren't always effective. And so that really was the motivation that, if I'm going to go into medicine, if I want to go into science, I want to also still satisfy my curiosity about it, but then also have an impact on my community. So that's really how I got into it. It's very, very late in college. I actually started grad school having never taken an immunology course, but it's on everything I learned just made sense. It was really a joy to learn. And so I just kind of just followed my passion in that.
Yeah, originally with immunology, it was because somebody had said that it was one of the hardest modules in medicine and in dentistry, and I didn't know if I wanted to do either of those, and so I decided to take immunology and fell in love with it. I agree with Michael. I like the domino effect of it. It made a lot of sense that everything was kind of connected, and very similarly, it was autoimmune disease for me as well. I've just really been interested in these diseases where the body is attacking itself, and we can barely tell the difference between different conditions, let alone treating them. And so that's why I like regulatory cells in general. So both B-regs and T-regs, I like them because I like this idea of they're a checkpoint that prevent like this overinflammation that's happening in your system. And so, yeah, that's to this day, I just really get excited learning about why autoimmune conditions and then food allergy, like on a micro level, like breaking tolerance, how that happens and why that looks so different between people.
Yeah, I find that quite interesting. You're making me miss allergy a little bit. I don't need that. I've just started a postdoc. So Following on, Michael, from your point where you mentioned racial disparities in the community in terms of treatment, we actually got a question from for Dr. Gadir about racial disparities in food allergy. Is that something that you see? Is that similar?
Yeah. So food allergies in general are like qualified, they're described as like rich people diseases, right? Because they're associated with like more affluent lifestyles, even though that's not actually true, because something that's commonly overlooked is that a lot of people with autoimmune conditions also have food allergies, right? So like, this was one thing about my PI, his perspective was, if you get a small break in tolerance in the gut, you have food allergies, and then systemic breakdown, you get all the like immunodeficiencies and like lupuses and whatever. And so it's definitely probably underdiagnosed in the Black community. But in answer to that question, yes, absolutely. Just in terms of the funding, and it's also a field that's heavily funded by like mothers. So there's a lot of like patient advocacy and mother advocacy. And so the types of money that's coming in and the types of communities that are recruited into the clinical trials, it's definitely a problem. But I know that there are a few people working on it on the front line to like increase diversity in the clinical trials, and then also start to look a little bit at the juxtaposition between asthma and food allergies. And asthma obviously definitely having kind of racial bias in terms of diagnosis and treatment. Trying to understand how school children develop atopy, going into like different school systems and trying to like recruit more diverse clinical trial subjects.
Mm-hmm. Yeah, no. And I think if there's one thing that the pandemic has shown us is what impact disparities can have on health outcomes. And I think it's not unique to food allergy. It's not unique to cancer. To cancer, we see it pretty much across the board. And I think that takes us quite nicely to what I wanted to ask you guys about being Black in immunology. Like, what has been your experience of being Black within immunology? Do you feel like you've faced any major barriers? And if you have, how did you navigate those barriers? Anyone can go for it.
I can start.
Yeah, go.
I think the lack of representation was the main barrier that I faced. And I think that sort of trickled down to a lack of understanding of certain limitations that we faced. So, it took me a while to kind of find my foot in the field because, we didn't really have, I didn't really have the guidance, and, there wasn't really anyone that I felt would relate to my circumstances that I could, talk to. And I specifically remember an incident where there was a platform and there was an opportunity for someone quite senior to talk about what, me and some other people were working on, and what the person had said. So the person had, a slide, and they had this unique qualities of everyone. And the unique quality that the person had put up for me was the ability to change my hairstyle. And I thought, well, this is a platform. You could have said anything else, but not my hairstyle. And what that had on me, the effect of that was that I started to keep exactly the same hairstyle every time, because I didn't want that to be what I was known by. So, it's that lack of understanding that sort of, it makes it hard for you to, you couldn't quite be yourself. You had to conform. And I think that was a challenge. But what I have started to do is take that as a positive, the fact that I am unique. And if I can do really good work, it would be harder to forget about me and forget about what I do. So yeah, that's my experience of being a Black person in immunology.
Dr. Okori?
Yeah. So I feel like my perspective is a little bit different because I started out in medical school and there are a lot of Black people in my class, and so I had a lot of friends and people that I could relate to. That definitely when I transitioned to grad school, I think there was only one other person in the program who was Black, and he happened to work on my floor, so we were able to to get a good relationship. And he was senior, so he really mentored me a lot through that process. So I felt like I was still supported in that way. So I feel like I've definitely been a bit lucky in terms of my experience in immunology. And then also Elaine, who's one of the organizers here and for Black Immunology, has been a great friend to me during the program as well. But I definitely would say that it can be isolating because there's just so few of us. And I recognize, one or two people not being there, then you're just isolated and alone. And then especially at conferences, it can be kind of tough, right? If you're the only person there, how do you present yourself and how do you, start a conversation can be kind of a bit intimidating. But for my personal experience, I feel like I've been very lucky because I've had a lot of help and a lot of support along the way.
Yeah, I would agree with that. It's representation, like the lack of representation has been really isolating, and it like sometimes you don't even realize like how much you don't think about it until you're at a conference and you see another Black person and you're like, whoa, hey, how are you? But I think it's that, and I'll say something that I continue to struggle with, and I think that a lot of people probably do as well. It's just how many people who are even within the field who don't understand how much racism touches every part of what we do. It touches like the diagnosis of the patients, it touches treatment of them, like it has like everyday real life implications. And I think something that I've always found really frustrating to navigate is being asked to compartmentalize politics and being asked to compartmentalize that away from my science. And I think it's something that I used to be much better at. I don't know if it's something that you're even good at, but now I'm finding it increasingly more and more difficult. And it's something that I spend a lot of time thinking about.
I want to hop right on that point. And I think it's impossible to do that, to separate yourself into different pieces. I think that your politics and how you feel in your field, how you feel racism is important, and the expression of that is important, and others should know that. They should know that these things make you feel different and isolated and alone, and those are things that are challenging to your success. And so I think representation is the biggest issue. No one stands in front of the door and says, no, you can't come in, you can't be an immunologist, but the lack of people who look like you and people who identify like you and understand you is challenging. And so it's really important to have a mentor, someone who looks you in the eye, who sees you and decides that I'm going to encourage you and look forward to your success and help you be successful. That for me is very important. And so I think all of us see ourselves as an example for others to follow, but how do you find that first mentor who does that? And that, I've been really fortunate to have that in my career, and I hope that you all have as well.
Yeah, no, I entirely agree. It is representation is the hardest part, but like I guess everyone here on this panel, everyone's very successful. Like you're all very, very successful people that people can look up to, and I guess the thing is like how do you feel like you've navigated? Is that through mentorship, like you said, or is it just the absolute love for immunology that's like pushing it all through? It's just like a mixture of all those things. Yeah, it's one of those. But yeah, so I guess we've touched on quite a lot of things. I want to go back to some more of the questions from the audience because they keep coming in. So one question for Dr. Gadir again is, what foods include the microbial communities that promote food tolerance. So are there any foods that we can naturally eat that have these communities in them, microbial communities?
The answer to that is I don't think that being exposed to the microbes is necessarily just coming in through the foods. I think these are events that can happen near each other, but not necessarily at the same time. I think there's a lot of data about early introduction of foods. So showing that if you introduce, similar to like oral immunotherapy, like if you introduce small amount, if you're allergic to peanut and you just introduce small amounts over time, that people will build tolerance. A lot of our data in the lab actually showed that with oral immunotherapy, you're actually not completely reversing the Th2 signature of the regulatory T cells, which implies that perhaps oral immunotherapy isn't able to like induce real tolerance. But in answer to that question, I think if you're a family who's high risk for peanut allergy, for example, because you have a lot of people in your family with peanut allergy, then you should probably introduce peanuts early or like keep an eye on that because that could help prevent. But I don't think it's as simple as eating kombucha and it'll have like the dietary antigens and the microbiome in it, for example. I don't think the story's that simple.
Yeah, no, that makes perfect sense. And I've got another question for Dr. Posey. Are there any large-scale applications of your technique?
Absolutely. So adoptive transfer of T cells that have been ex vivo manufactured into large numbers, either unmodified tumor infiltrating lymphocytes or genetically modified TCR expressing T cells, CAR expressing T cells, and other therapeutics that may come further down the line is happening now clinically. We have three FDA-approved CAR T-cell therapies. These are all currently personalized. They're all autologous T-cells that are removed from the patient themselves, manufactured, and then reinfused into that patient. But we will see, we do see clinical data in clinical trials, and we will see in the future allogenic CAR T-cell treatments. We will see T-cells derived from induced pluripotent stem cells as a bank of cells, and so those applications are happening. There are, I believe, over 100 companies doing CAR T-cell or T-cell immunotherapies now, and what we think is that these glycopeptides that we're interested in, these abnormal glycans attached to a particular peptide, is a way to differentiate a tumor cell from a normal tissue, and will increase the sensitivity of these therapeutics and hopefully lead to some good results.
Yeah, that's cool. And following on to that, is there a TNome?
That's a great question. The answer is yes. There are ways, and there are groups in the world who study O-glycosylation sites on every protein in the proteome, and have identified through generation of TN expressing cells where proteins are O-glycosylated. We do some of this now. We use the Jurkat cell line because it naturally expresses TN antigen on all O-glycosylation sites. And so if you're interested in whether or not your protein of interest is O-glycosylated, you express it into Jurkat cells, and they will include TN antigen on that particular O-glycosylation site. It won't get elongated any further, and so you can enrich for those TN positive peptides, and we're interested in identifying more of these peptides in different tumors.
Okay, very cool. Yeah, and I've got maybe time for maybe one more science question. So this one's for Dr. Okori. Oh no, I've actually already answered that question. So in that case, I'm going to actually ask the question I wanted to ask. So I was going to ask you, so you see CXCR4 expression, and you're saying that there's a function in the development of early B cells. Is there any function of it in mature B cells? So once you're already out and you're inside your spleen or your respective organs?
Yeah.
outside of chemotaxis?
Oh, outside of chemotaxis. That's a
Yeah.
good question. Not that I'm aware of. I know that CXCR4 is important for transitioning between the light zone and the dark zone during the germinal center reaction. I think we're also pretty interested in the lab, given what we know about CXCR4 in early development. Maybe those signals are also involved in the processes of, essentially recombination and affinity maturation that's occurring in the germinal center. Maybe those sort of signals are also important in the germinal center as these cells are going to light zone and back to dark zone, and affinity maturing in that process. So there's potential for it, but I don't think there's any data out there right now.
Yeah, I think that'd be really, really interesting. So now that I've pulled us back to the science, I just thought I'd ask, so if, well, obviously we're going to, we're all Black in Immuno, we're all doing amazing science. I'm including myself in this. I don't know why I haven't told you any of my science. But if we're all doing this, how do you think we can improve our connection as with our communities to rebuild trust within science in general? And I'll pose this to Dr. Gadir.
So I don't know if anyone else on the panel would disagree with me, but I actually think it's really important to acknowledge all of the kind of some of the mistakes that like modern medicine has made. I know that there's some data that shows that if you apologize, it doesn't actually mean, people always throw this around, but I think that acknowledging it would do a long way. I mean, my experience is that particularly in my own communities, it's not that people necessarily have mistrust in medicine, which means that they completely discount it and are unwilling to grasp science. They just want to keep a level of healthy skepticism because history would tell them that it would be silly not to. And so I think personally, one way is increased education around things that have happened in the past, and more education around how that's changing, and increased visibility.
Yeah, that makes perfect sense. And just to close, I'll just ask a question to the whole panel. So if you had to give one piece of advice to a young Black immunologist coming up and emerging behind you, what is that one piece of advice that you would give them to take and kind of excel? So I'll just go in order of people I see. So I'm going to start with Dr. Posey.
Yeah, resilience, work on your resilience. Be tough enough to know that a failed experiment, or a week's worth of failed experiments, or a month's worth of failed experiments, or a whole year, it will not stop you from succeeding. Keep going, keep pushing through.
Oh my gosh, you don't know how much I needed that advice right now. I'm going. So Dr. Ogbe, what about yourself?
I would agree with exactly what he said. It is resilience all the way. You have to be able to bounce back after every failed experiment. And yeah, I would say the same thing.
Okay, cool. Dr. Corey?
I would say to be absolutely intentional about the mentor that you end up doing your PhD with. So not only is the science important, but your personal relationship with them. Is this someone that you can actually let your guard down and talk to about things other than science? Let them know if you're feeling insecure about, where you are in terms of your understanding or your presentation skills, whatever. There needs to be somebody that you can fail in front of, and they're going to be willing to pick you up and show you the ropes on how to do things better. It shouldn't be somebody that you're intimidated by. It shouldn't be somebody that is going to, demean you or anything like that. So be very intentional about your PI, making sure that you have a really good personal relationship with them, because that can honestly be make or break sometimes in graduate school.
I agree with that, and I would add pick a PI who loves what he does, or what they do. I think it's having mentors that really loved immunology made me really love immunology. So I think the other part of it is to make sure that you pick a PI who has a lot of ideas and really does enjoy kind of sitting and brainstorming, and gives you time for that, because that's the piece of it when the experiments aren't working. Having a mentor who has good ideas, who keeps you excited is really important.
Yeah, no, I 100% agree with that. And I'll just like to take this moment to thank all of you for your talks, for your insight on this panel at the end. And I personally really enjoyed all this adaptive session. And thank you for participating and getting involved with Black and Immuno Week. So Black and Immuno Week is going to continue. We've got more events for the rest of the week. Please get involved with Twitter. And I think if your questions haven't been answered, they hopefully will be answered via Twitter. But thank you for